that, let's get going. I would like to turn it over to the president of the Children's Tumor Foundation, and that is Annette Bakker. Annette, please take it away.
Oh, good evening, everybody. And I would really like to thank everybody who is taking the time tonight to participate in the update on the NF2 Accelerator. You will observe that we in the Children's Tumor Foundations have really chosen for a portfolio approach. And for the investors on the call, portfolio is a way to balance risk and reward with the end goal to really maximize the return on investment. And as you know, our return on investments is really to bring drugs to the clinic for clinical trials. For the non-investors on the call, I would like to present it a little bit different. It is a little bit like the race you're seeing here, where instead of betting on one runner, the foundation has chosen to bet on multiple runners. That means, in fact, practically, that for the moment we are betting on drugs that are out there so that we can help the NF2 patients today, while also investing in the, let's say, unknown holy grail, which is NF2 gene therapy. So tonight, it is my absolute pleasure to introduce you to Salvatore LaRosa, who is our Chief Scientific Officer, to give you an update of what the initiatives are that we have. And we also are very fortunate to have two guests on the phone tonight. One is Dr. Plotkin from MGH, who is the principal investigator of the brigatinib clinical trial, and he will explain you where we stand and where we go. And the other person is Mr. Michael Wootten, who is the Chief Executive Officer of the company called NF2 Therapeutics, and will give you an update on the NF2 gene therapy efforts. Towards the end, Michelle will give you an update on where we stand with fundraising-wise for the NF2 Accelerator. And with that, I really would like to thank everybody for being here tonight, and we're hoping to get a lot of questions from you. So with that, let me turn it over to Salvo to give you an overview of where we stand.
Okay, thank you, Annette. I hope you guys hear me. Just getting confirmation, yes, from the captions. So as you see here in the overview, we'll cover the first 3 points that are the main pillar of the NF2 Accelerator Initiative. And I would like to explain a little bit about the clinical drug pipeline. These are what Annette said. We are trying to push drugs that are easy to be translated into treatments. And we have some front runners with brigatinib, and Scott will soon talk about this. And then we are trying to make sure we are making investments to bring the next drugs in the clinic after brigatinib. And then after that, we're definitely trying to fuel new ideas and new science for NF2 treatments as well. Then the second point is not only having drugs, but also having a system in place that will allow us to select drugs that have better capacity, better chance to get to the finish line, to get approved by the FDA. And here we are working with the experts to leverage some of the technology, for instance, with large animal models. And I will talk about the mini pigs initiative we're funding for NF2 and other models that we want to make available to all researchers. And then at the end, Michael is going to talk about the recent investments we're doing with NF2 Therapeutics and their approach to gene therapy for NF2. And then, of course, some important dates, fundraising, and Q&A. So let's go to the first runner, the brigatinib clinical trial. So what I want to do is, first of all, explain you briefly, where is this drug coming from? And then I will pass the mic to Scott, and he will actually talk about this trial, where we are, and all of the details about it. So where is this drug coming from? Brigatinib was identified in Synodus NF2. And for those of you who know and heard about this project, it was started in 2014, and we invested 3.3 million. CTF had a pivotal role in this, not only in pulling this initiative together, but also coordinating it. And it was a major effort. 12 labs across the world, and there were 12 labs, 8 different institutions. So if you see here on my bullet points, the program was completed in May 2018, and these are the major results. So we tested over 2,000 drugs. We also tested, out of these drugs, 170 different drug combinations, meaning 2 drugs together. And we saw that there was a clear advantage of testing combinations versus single drugs. Then we'll look at, even more, like 10 drugs to see what was the effect. And if you see on the right of your screen, it's like, taking a funnel where you have all of this drug at the beginning, you funnel through your system that is making more and more selective and selecting always the best. And at the end, you have a potential therapeutics, potential drug. So that's where brigatinib came out. Of course, there is a lot of other data there, and we are trying to see what is next after brigatinib. But this is clear that the #1 results of Synodus NF2 is this drug, and now we are about to enter a clinical trial with this drug. So I will turn to Scott, and he will talk about the paradigm for platform trial, current status, and the next phase. So I'm gonna stop sharing my screen. I know, Scott, you want to share your slides, right? We cannot hear you.
I'm not seeing Scott. Did he?
I don't see Scott either.
Yeah, I wonder if he got bumped off. We know he's here before, but no, I don't see Scott here.
Okay, can you unmute Scott, because I think he's muted?
I'm looking for the name Scott Plotkin, and I don't see him. Is he logged in?
Yes, he's logged in.
You should.
ID 67.
Albert, do you see him? Salvo, you should be able to see it too, because you're.
I don't see him either in the attendees list either. It's almost like he just disconnected somehow.
So just for everybody, I have Scott on the phone, is logging out and logging in again. Just a few minutes, sorry.
Okay.
I'll keep an eye on him and promote him once I see him.
There he goes.
Can anyone hear me yet?
We can now.
I could hear, remember, I was talking to Annette in the beginning, so that's why it was confusing. So I apologize. Let me.
We've got you, Scott. Go ahead.
Yep, we promoted you to presenter, so you could go ahead and share your screen.
I will in 1 second. I want to make sure the captions are working for me also. I see them. Okay. Then give me 1 more second to figure out how to show my screen. Share screen. Can anyone see my screen?
Yes. Look.
Yes, we see it.
We see it, Scott.
Great. Well, thank you very much for the opportunity to provide an update on all of our work. I do apologize. I've been sick the past 2 days, and I probably sound like it and will be a little bit feeble, but I'll do my best. This is an update that I also provided to the Department of Defense NF Consortium recently. So this is the same information we provided to investigators. So as mentioned previously, this work comes out of the NF2 Synodos project and is a collaboration between Children's Tumor Foundation, Takeda Pharmaceuticals, and a group of investigators. And what I'm going to tell you tonight in a few minutes is about an innovative trial design that incorporates the use of the drug brigatinib. But I want to emphasize that this trial is far bigger than any single drug, and I hope to convince you of that shortly. It's the main reason that it's taken us some time to get this up and running. So this trial is called the Innovative Trial for Understanding the Impact of Targeted Therapies in NF2, and we're going to abbreviate it into it NF2. And just before I start, can you remind me, Salvo, how many minutes so I can time myself here that I have? Or you want me to speak?
Just take the time you need, Scott. We just keep in mind that we're kind of late already, but anyway, you take the time.
So this is a brief table, or a limited table, I should say, of many of the clinical trials that have been run in the past decade for NF2 related tumors. And one of the points that I like to make is that for clinicians and for patients, our development of drugs in NF2 has been incredibly slow. And there are many reasons for that. One of the reasons is that it takes years to get a trial through the regulatory process. And we're looking for better designs to make that faster. So this is a summary of some of the challenges and opportunities for NF2 patients and clinicians. We know that current therapies such as surgery and radiation have worse outcomes than for sporadic tumors, meaning similar tumors in patients who don't have NF2. Everyone is aware, of course, that there's no approved therapies for NF2 right now. And the point that I'm trying to make tonight is that the time between trials is way too long. But a second important problem is that when we have two trials, it's difficult to compare the results to say drug A versus drug B. And that's because different trials have different designs. And for that reason, the results may not be comparable. So I want to tell you about some of the work that's going on in oncology more broadly, where we're designing new trials that answer or address some of these questions. The first thing I want to explain to you is the concept of a basket trial. And this basket trial tests the effect of one drug on a single mutation in a variety of tumor types at the same time. So for example, we know that some types of lung cancer and some types of breast cancer can be caused by the same genetic variation, even though the cancer types are very different. And in a basket trial, you could take drug A and treat a breast cancer with a mutation and a lung cancer with the same mutation in the same trial. So instead of the traditional one drug for one tumor type, a basket trial allows you to enroll multiple different tumor types that share a common cause into a clinical trial. And we call it a basket trial because all of these different tumor types get placed in the same basket. A platform trial is a trial design that incorporates not multiple tumors, right? That's the basket is multiple tumor types. A platform trial is something that has multiple drugs at the same time. And the way a platform works is that individuals get randomized or allocated to different drugs. So the nice thing about a platform trial is that all of these drugs are provided to participants using the same eligibility criteria. That is, they all have the same reason for joining the trial. And so it promotes comparability or comparisons between the treatment arms. So these two designs are being applied broadly in oncology now. And our goal in the Intuit NF2 trial is to combine this platform and basket models into a single trial so that we can take the broadest range of NF2 related tumors into treatment and use multiple drugs to be screened in sequential fashion, that is, one after the other. And platform trials by design are flexible. That is, if one of the drug arms or the experimental arms doesn't look good, it can be stopped. And if new drugs become available, you can add them into the trial without having to start a whole new trial. There's an expedited way to add these trials. And what it requires to do this is a pipeline. And that is the work that Children's Tumor Foundation, along with investigators, have been doing with Takeda for the past 2 years. That is, not focusing just on drug A, but focusing on trying to develop a relationship with a major pharmaceutical partner that can fill our pipeline of promising drugs for the next decade. And here's a schematic of how we envision this working. This slide is a reminder that I'm presenting you our design, but this is likely to evolve. So if you are viewing this beyond 2019, there may be changes to this trial design. With that understanding, I'll walk you through it. Salvo, can you see, or Simon, my pointer moving around?
Yes, we can.
Okay. In the top left, you see the platform trial. The portion shows that patients have common eligibility, so the same types of reasons to get in the trial, and we evaluate the success with the common endpoints. So this is going to encourage comparison between the drugs. Now, patients with NF2, in order to enroll, have to have a tumor that's growing. We're not going to provide drugs to individuals who have stable tumors because we don't want to incur side effects without benefit. One of the novel designs is that this is a basket. So instead of only focusing on vestibular schwannoma, this trial will allow us to enroll vestibular schwannoma patients, patients who have growing non-vestibular schwannomas or growing meningiomas, or for the first time ever, growing ependymomas, spinal tumors. Individuals will be randomized, which means like a coin toss, to either receive drug A, drug B, or drug C. These are called individual drug substudies. So the top box is what we call the master protocol, and the box below are the individual drug substudies. Now, one thing that's important is when you start off with only a single drug, such as brigatinib, our first drug, there will be no randomization. But in the future, as we build our pipeline, patients can be treated with a number of drugs at that time. After being on the study, we will look at the MRI scan to see which tumors are responding and which not. If somebody's tumor remains stable or shrinks, they'll stay on the individual drug substudy. If the tumor grows, what we see then is that this individual comes off of that individual substudy, comes back into the master protocol where we will observe them, if they choose, by getting routine MRI scans until there is evidence that the tumor is growing, and then they can potentially be randomized a second time to a different drug if available. So imagine a scenario where there are 3 drugs. If a person's on drug A and the tumor grows, they come back into the master protocol. Once the washout period is done, because there has to be a wait time so the medicine leaves the body before you can start a new medicine, you can immediately get randomized to the second drug. And so we hope that when the trial is mature, that the wait time between experimental agents is somewhere between 3 and 6 months. And the goal is to give patients as many opportunities with promising drugs as is possible. I want to stop there for a moment and just see if there's any obvious questions. And I can't see the chat. So Simon, could I ask you to just, if you see any questions, could you relay them to me, please?
Sure. So we have a question from Ann Noble, and she's asking, do they know what any of the side effects are with any of those?
Yes, we have I can only tell you about the first drug so far, and I'm going to tell you later in the talk, so why don't I wait? But I will say that this drug, brigatinib, is FDA approved for treatment of patients who have lung cancer, and we have a lot of information about the side effect profile, which is mostly nausea, vomiting, rash, swelling, and things like that, but generally well tolerated.
A couple other questions, one from John Morris. Are the drugs only coming from Takeda?
At this point, drugs are only going to come from Takeda. The nice thing about working with a large pharmaceutical company is that they have an enormous pipeline. I think our vision moving forward would be to have a trial that incorporates multiple companies, but being realistic, we know that there are intellectual property issues with trials, meaning that companies tend to want exclusive rights. And so it is a challenge in some cases to work with multiple companies, but that would be one of our future aims if possible.
And a question from Barbara Franklin. Will the initial basket include 3 drugs, which, hang on one second because the chat's moving. Will the initial basket include 3 drugs, which will all treat VS or MEN or EPEN?
So remember, the basket is a term for the multiple different tumor types. So all 4 tumor types are eligible from the get-go. The first drug, there will only be a single drug when we start, and that'll be brigatinib, which will be drug A. But all 4 tumor types will be eligible for treatment.
And a follow-up from Barbara. What if a major pharma company has a very logical drug? Will there still be clinical trials for it?
I'm sorry, say one more time.
What if a major pharma company has a very logical drug? Will there still be clinical trials for it?
Sure. It probably, at this point, on December 11th, 2019 would not be the Intuit NF2 trial, but we are still open for business at every institution and in the DoD NF consortium to run trials. We would never turn away a promising drug.
And I think Ann Noble said, as a follow-up to the side effects question, she said she meant more along the lines of total fatigue.
That's a good question. I would say every drug I've ever worked with has fatigue. And so I'd have to look back in and really look back on the specifics for bergatinib to see if it's out of proportion to other medicines.
Okay, I think that's all the questions for the moment.
Okay. Now, given the amount of time we have, I'm going to try to focus on areas that I think are most productive. This is a list for the doctors on how we're going to assess whether the drugs are active or effective. And I'll just point out that in addition to looking at the shrinkage on MRI scan, we're going to be looking at safety and tolerability, of course, hearing response for individuals who have measurable hearing, quality of life, and we're going to be looking at a variety of biomarkers, meaning looking at archival or stored tumor samples, as well as blood assessments on genetics. So as of today, subject to change, the way to get into the study, these are the inclusion criteria. We an individual has to have a diagnosis of NF2. Excuse me. The company did not want us to treat anyone under the 18, but because we and Children's Tumor Foundation have been very strong advocates, we have been able to explain to them the importance of treating individuals younger than 18. So at this point, we're open age 12 and up. And the reason it's not below age 12 is many of the medicines we're studying do not have safety data in the young children, and therefore the companies are not prepared to do new risk in this setting. I mentioned that tumors have to be growing. There's two possible definitions. It's growing over the last 36 months, either a 20% increase in volume or 2 millimeter increase in linear dimension. The tumors have to be measurable, which means that you have to be able to see them on MRI. Patients have to have a life expectancy of more than a year, meaning you can't be so sick that we worry that you're not going to do well. And a performance status over 70%, which means for most people, the ability to at least do some walking. That's a rough idea. You have to get the specifics at the time of enrollment. And the exclusion criteria, if you're on chemotherapy or getting radiation, we don't mix medicines. Radiation within the last 3 years, because it confounds or confuses our ability to understand who's responding or who's not. You have to be able to tolerate MRI scans. No major illnesses, and pregnant or lactating women are not eligible just because we don't know the effect of these medicines in that group of individuals. So I'm going to, in the last few minutes, just tell you about how many individuals we hope to enroll. On the master study, we want to have 80 individuals, and on each individual drug, we hope to have 40. And these numbers are easily changed. So these are just what we're using to start things off. The statistical plan is modern and flexible. It's what we call an adaptive design. And so the way that adaptive design works, that patients will be enrolled in 2 stages. In the first stage, 20 individuals with any of the tumors we talked about will be accrued or enrolled. We want at least 2 people per tumor type. So 2 people with vestibular schwannoma and 2 people with non-vestibular schwannoma. After 20 people are treated, we're going to look at the MRI scans, and we're going to try to figure out, of the 4 different tumor types, which one looks the most promising. Maybe the drug works really well in meningioma and works terrible in schwannoma. Or maybe it works great in schwannoma, but not in ependymoma. We will then take that information, take 2 of the 4 groups, and the last 20 subjects, the second 20, will be accrued only into those 2 baskets, because we are trying to maximize treatment of people whose tumors are likely to respond to the medicine. I won't go through a power statement, and I guess I won't go over the Synodos NF2 work that we talked about previously. Since Salvo did that, I'm going to see if there's anything else.
No, Scott, maybe you can address when is the trial likely to start? I think is a question from Aaron as well.
I see. Yeah.
Yeah, in fact, we have a few questions after you answer that. We'll throw a few others at you.
Okay. Well, so the stage we're in now is we're undergoing ethics review, which is, for those who are following the details, that's an institutional review board, or IRB. Each institution has a different protocol or way of reviewing these. At Mass General Hospital, which will be the coordinating center, we have 2 stages, and we're in the middle of those right now. And so I think that we're hoping in the next month or 2 that we will be able to start enrolling patients. That's our hope. Christmas is always a very challenging time of the year to get things through these regulatory groups.
Another question from Aaron Lipoff. Can Scott please speak to any off-label usage of brigatinib for NF2 and outcomes for the same?
I'm not aware of any published data on the off-label use. This is always a good question, because we're going to have a choice as a community. Are we going to enroll patients, we the doctors, and our patients going to be willing to be enrolled on the clinical trial? Or will people prescribe this or take this off-label? My hope is that we will have enough individuals who will go on the trial, because that will be our only way of formally learning whether it works. So I don't know what's going to go on in the community, but I hope that we'll have enough individuals that will be on the trial.
Next question from Barbara Franklin. Will those in trial have to go to specific centers or local hospital?
So, like all clinical trials, the United States government regulates them very carefully. And as part of that, each site has to have federal approval to be part of the trial, which means that your local doctor can't just do this. So the answer is there will be sites, and we're trying to arrange it such that they are spread across the United States. But we all know that there's never enough sites to be convenient for everybody. And one of the things we're trying to do is to make this to make it easier for people who travel. And if we're able to get it through the regulatory bodies, there may be ways of doing 3 months between visits in some cases. So I'll keep you posted on that, depending on how it turns out through the ethics board.
Scott, just to make sure that we are all and that we don't raise expectations, in a month we will be submitting the protocol to the FDA, right? It's not that in a month we will be able to enroll the first patients. Just to make sure that we're all not raising expectations, where I want to make sure.
Yeah, I mean, I'm struggling with an unknowable answer, which is people understandably want an answer. And on the other hand, you and I, you being CTF, me being the MGH investigation team, we don't control it. So you're right, absolutely right, Annette, that I don't think we'll be enrolling patients in a month, but I don't know where we'll be. It will almost certainly be longer than that, but it's hard for me to know the exact time.
Another few other questions. This is related to this. I don't know if anything to add, but Rima Bogan asked, when does the phase 2 trial for brigatinib start? And from there, if the results are promising, when will it be available as a treatment method similar to how Avastin is used now?
Well, it's an FDA-approved drug, Rima, which means that anyone can technically prescribe it, but that does not mean insurance will pay. And I already stated that my hope is that we, as a community, try to enroll enough patients so that we get an answer here. Having said that, I think Annette's point is well taken, that I don't want to come across as too optimistic that we'll be enrolling in January, because she's right. That's not likely. So, you know, I think first quarter is our first quarter of 2020 would be my hope. But you never know what you run into in your review. And so it's hard to know for sure when first patient will be allowed to start.
From John Morris, what about patients with multiple tumor types? I thought that was common in NF2. What basket are they in?
So it's a great question. And the answer is you have to have a growing tumor. So if there's one, we call these target tumors and non-target tumors. And so it'll be up to the clinician and to the individual how to define this. So if an individual has a growing schwannoma and a growing meningioma, they can choose. If they only have one type of growing tumor, that would be the logical choice.
We've got quite a few questions. From RB Harrison, what sites have been identified to date?
So we have done a competitive application process with the goal of identifying sites that see large numbers of NF2 patients and have the infrastructure to enroll patients to clinical trials. That selection process is not entirely complete yet, in that we do have an idea of who it's going to be, but we have not negotiated budgets. And so until sites are willing to accept budgets, it's not finalized. So until the process is finalized, I don't think it's going, or near finalized, I should say, we're not going to be discussing individual sites unless Annette feels differently.
No, I think it is careful not to give the names of the site yet. We are discussing, as you can imagine, everybody wants to participate. It's a little bit a competitive trial to participate in, and we want to make sure that those sites that can recruit the patients as quickly as possible will be the ones that will participate in this trial. And we also don't want to include too many sites, because then it becomes just a nightmare.
2 or 3 more questions. Lisa Lewis is asking, will any sites be considered / approved in Canada or only the US?
At this point, it's, to my knowledge, it's the US, and there's a lot of reasons for that, which is I have tried personally to open trials in Canada and in other countries. It's incredibly difficult and cost millions of extra dollars. So at this point, I'm not aware of any possibility of doing in Canada at this point. I mean, I don't know if you feel any differently in that, but you need incredible amounts of infrastructure.
Yeah, no, I agree.
Question from Michelle White. If I'm supposed to start Avastin in the next week, would that disqualify me, or would I be able to come off of it and still participate?
You could, so what you can do and what you should do are 2 different questions. So obviously this is something to discuss with your treating physician. But there will be allowed a washout period for people to start. So it would not disqualify. There's not, in some kind of clinical trials, there's a if you've been treated in certain ways, you can't receive the drug of interest. That's not the case here.
I think that's it for now. There was one other question, but I'm going to put that towards the end since we were focused on clinical trials at the moment. So, and also keep in mind that it's 9 o'clock. So.
Okay, sorry about going long.
No, I think this was good. But Salvo, I'm going to let you sort of take lead on the next piece.
So if you can stop sharing, Scott, I will take from there. Or I think I can, yeah. Okay, I'm going to take from where I left. And as you can see from this slide, I'll go faster from here because I want to give Michael enough time to talk about the gene therapy that I think is very exciting. But what I want you to see in terms of where our project is and how our fundraising initiative and money is allocated, that I put the price tag of fundraising target for each one of these initiatives that is listed under the NF2 Accelerator. And we have managed to secure $350,000 for the brigatinib trial. Of course, this is not covering the entire budget because I think Takeda Pharmaceutical is taking a bigger portion of this, definitely bigger. So it's going to be a co-funding, and CTF is helping to fund, the money needed to, let's say, cover the cost of the PI. So that is absolutely necessary for this to happen. So it's going to be $350,000 that we are initially allocating, and we're going to spend as soon as the trial is going to start. Okay, so just want to let's see, go back a little bit and tell you how we are planning to move forward, because it's true that brigatinib is the front runner and is ready to go, and probably next year very soon we'll have it in first patient's dose. That's what we hope. But what can we do to create that pipeline that Scott was mentioning? Now that we have a platform that can accommodate multiple drugs, now the next need is to fill a pipeline where we can actually create more drugs opportunity. So new drug testing. So we want other drugs in clinics for NF2. And how do we make sure we have a pipeline of option? First of all, CTF is looking at different opportunities. We launched recently a request for application, or RFA, for new NF2 drug testing proposal, and funded 3 groups recently. One is the group of Jaya Ramesh at Massachusets at MGH, where she's actually taking a second look at the Synodus NF2 data that contains a lot of data. And there are new drugs combinations that have, the capability to be the next drug after brigatinib. So this, we just funded her work, is going to be testing in animal models. And then if this is promising, we'll make sure that, it will get to the clinic. Also, we are funding a group in Spain from Elizabeth Castellanos that is looking at different drugs, antisense oligonucleotides. These are, innovative drugs that have promised to act differently. I'm not going to the specifics, but it's very interesting to see. It's very early, a little bit early, because this is going to be only in cells, and then we will need to move it into animals and then into clinical trials. But it's a completely different angle that will give us an idea on the technology. And then we are also funding Chun-Ling He at Georgetown University with a DDI, where she is looking at a pathway called HIPO-YAP-TADS. And these inhibitors are very active for NF2 in cells and animals. So she's testing specific drugs to see if the effect is what we actually hope for. And that will be a smooth transition into the clinic in the next year if this is available and working. In addition to this, we are open for receiving other applications in our Drug Discovery Initiative Registered Reports program. It's currently open, and we're waiting for application until the end of the year. And next year, we also will launch new RFA for more drug testing proposal. The budget on this part is going to be $640,000. This is our fundraising target over, you know, the whole initiative of the accelerator. And then, in addition to this, if you think about drugs, we also want new ideas, and new ideas are coming from basic science research. So we are adding to the drugs that are already there, that are ready to go into animals and humans. We are adding a component to create new discoveries, to look at different mechanisms, to fuel new research. And our Young Investigator Award program is the perfect program to start these new discoveries. So researchers have access to experienced enough mentors and labs to explore new science. We have now open for proposal until January 4. And what is really important is this is the first year, because of this initiative, because of your support, that we have an NF2 restricted budget set aside for this program of $640,000. And this is going to fuel also the next wave of investigators for NF2, because giving a grant to a young researcher has a very high chance that will stay in the field and, you know, will establish his career with working on NF2, we hope. So moving forward, I'm going to talk about how the other part of the proposal of the accelerator, that is improved drug selection and NF2 models. And as you can see here, the price tag that we put on is $420,000. And some of this money has already been spent, for instance, in large animal models for NF2, and this is what I'm going to talk next. As you can see on this slide, there's been a little bit of work, I would say, already done by CDMRP, which is the Congressionally Directed Medical Research. We gave a grant to University of Minnesota and Recombinetics, which is a company that is specialized to create engineer mini pigs, genetically engineered mini pigs. You can see here, this is a mini pig, the founder of an NF2 colony that has been genetically engineered to develop NF2 the same way as patients are doing. So this is not any artificial manipulation. Is born with it as any person with NF2. And Marco Giovannini has received a grant from us that, you know, is looking to see if these animals will develop NF2, and is going to use this to understand etiology, disease development and progression, and the application of novel imaging and monitoring technique. As you know, swines and mini pigs are large animals. They're they're closer in terms of, you know, to the physiology of humans, more than, you know, mice or rats that are normally used for, you know, drug testing. So this new model will definitely help us to close the gap and maybe have better model to test those, you know, do new testing. And the other things that we're trying to do is to increase the testing capacity for NF2 drugs. What we see is that, there is sometimes it takes a very long time to create the opportunity for companies, but also for other researchers to access NF2 models. Annette, myself, we are receiving a lot of calls nowadays from companies, but also from other researchers. They want to test drugs, and sometimes these models are, exchange, or this contract that needs to be done, they're not as easy as we would like to be. So we are working with Wake Lab and other experts, NF2 experts, to create these models and make it available for everybody who wants to test a drug for NF2. I think this should not be the bottleneck. The bottleneck has to be the number of ideas that we have, but not the models that are available to test that idea. So it's mainly 4 different stages. We are at the first stage of generating the models, and then we'll move to validate the phenotype, establishing a testing protocol, and keep colonies alive and available for those who want access to the model. So that will create opportunity for everybody to test new drugs. And then it brings me to the NF2 gene therapy. My pleasure to introduce you to Michael Wootten. He is, I think, the CEO of NF2 Therapeutics. Can introduce himself. It's been a great pleasure. We've been working with Michael since many years. We also have a collaboration for NF1. And Michael, I will leave this slide on, and you can talk through the project that we are undertaking.
Thank you very much, Salvo. So this is Mike Wootten. I am the CEO of NF2 Therapeutics. And the company was founded by 3 individuals, myself, Scott Plotkin, who you've already heard from, and Mark D'Souza. As Salvo mentioned, the 3 of us founded another company called Inflection Therapeutics. And Inflection is focused on NF1. And Inflection is developing a topical MEK inhibitor for cutaneous neurofibromas in NF1, and has moved very quickly in that development program, and will be in the clinic in 2020 with trials in that program. So we're very excited to be focused on NF2. And the NF2 Gene Therapy program builds on some work that was previously funded by the Children's Tumor Foundation. And that previous work, we were looking at if we, using brute force, restored the NF2 protein, if we restored Merlin to a cellular model of the disease, could we restore function? And we worked with Dr. Helen Morrison, and what she was able to demonstrate in her lab was that if the Merlin protein was restored, function was restored in a cellular model of the disease. And so that was a very exciting discovery, and it was the basis for the gene therapy program that we are undertaking. I'm getting some feedback. Can people hear me okay?
Yes, Michael, you come across very clearly.
Okay, thank you. So the focus of NF2 Therapeutics is to develop a disease-modifying gene replacement therapy for NF2. And we are developing an AAV-based gene therapy which will restore Merlin. And essentially, for folks who may not be familiar with gene therapy, what gene therapy does is hijack a virus and engineer that virus so that its normal behavior of infecting cells is leveraged, but rather than taking over the cell and forcing a cell to produce copies of itself, the virus takes over the cell and forces the cell to produce copies of a protein of interest. In this case, the Merlin protein. And the gene therapy is AAV based. AAV stands for adeno-associated virus. And the reason for that is that AAV based gene therapies have been involved in a number of approved therapies, and they have been safe and effective. So they have not had significant toxicities associated, and they have been disease modifying, so life-changing for the therapies where they have been approved. Another sort of attractive factor of AAVs is that they don't incorporate themselves into the genome of the individual. So they don't modify the genetic material in the patient's genome. They will not be passed down to subsequent generations. The virus attaches it to the cell. It is taken up by the cell, and then the genetic material goes to the nucleus of the cell, but it does not incorporate itself into the chromosomes of the cell. And so our current activity, we are synthesizing vectors, and we will by the end of the year have the vectors, so the virus with the payload that we intend to deliver, and we have 2 studies that we are planning for 2020. One is a non-human primate study, and so that study will be in cynomolgus macaques, so in monkeys. And the reason to work in monkeys is that with gene therapy, it's important to have a model that is close to humans. And some of the other model systems that have been used in other research programs don't translate well to the human experience for gene therapy the way that non-human primates do. So that's the rationale for that study. And the non-human primate study is really going to be looking at a number of things. One is toxicity, and first of all, toxicity of the virus. And I should say that one of the other appealing factors of the AAV virus and why it's used for gene therapy is it doesn't create illness in humans. So humans can be infected by the virus, but there isn't there's not a disease associated with the virus. And however, for approval and to develop a new drug, you have to demonstrate that there is no toxicity associated with the virus. And so that's the purpose of this study. We'll also be able to understand if there is toxicity associated with the overexpression of the protein. So we'll have overexpression of the Merlin protein, and we'll be able to understand the impact of that in the non-human primate study, as well as the biodistribution of the AAV, and specifically if we are seeing transduction in the cells of interest. And that's really what's vital, is that we confirm that the virus is being taken up by the cells of interest, so that we know that we are getting the protein into the cells of interest, so that we can restore function. The second study that we're planning for 2020 is a mouse study, and this is a transgenic mouse. It will be in the lab of Dr. Marco Giovannini, and in that study, we'll also be looking at toxicity and biodistribution, because his model is a disease model of NF2. We will also be looking at tumor regression. So we're very excited to be embarking on this work, and love to have any questions that folks have about the gene therapy program.
If any, we do have questions that go back to the Synodos earlier, but if anyone has any questions for Michael, please do type them in. Okay, I see one from Tawny Pendleton. In addition to looking at restoring Merlin function, has anyone looked at the 2011 study, missense mutations in the NF2 gene result in the quantitative loss of Merlin protein and minimally affect protein intrinsic function? That studied two different molecules that significantly increased Merlin half-life, slowed the degradation of Merlin.
Yes, Scott, I don't know if you're still on with us, if that's one that you want to speak to.
Oh, okay, good. I can unmute myself. Yeah, thank you. So I'm trying to remember off the top of my head that manuscript, and I have to admit I don't. I'm certainly happy to look at it. I guess my first thought is just to say that there are many approaches one could take to restoring gene function or protein function. And I think at this stage, we wanted to take the approach that we thought was the most likely to lead to information that would be helpful. There are many ongoing gene therapy programs where replacement is the goal, such as in SMA or in hemophilia. And so we wanted to take what we thought was the least risky, most likely path to get an answer. So I can look back at that 2011 study, but that was at least my rationale. And Mike, feel free to add if you want, but that's how I think about it.
Yeah, certainly. The relative to small molecules, the development timelines have sort of tended to be longer with gene therapy development, and we certainly our strategy is to take a path that gives us the best chance of getting something that would be clinically relevant as quickly as possible. And Scott, feel free to add in any other thoughts just on the overall gene therapy program.
I thought you did a great job. So really great. Let's see if there's more questions. I'm looking up this.
Tawny responded, said, thank you for the answer. One of the molecules was a naturally derived botanical quercetin.
Oh yeah, I'm familiar with those. Yeah, I would like to see more replication of that data before we would put in a big effort, but I know what you're talking about now. I'm gonna look it up.
So this is Annette. Maybe one reply to the mutation paper is that although those are things that we don't know, right? If you have a functional or a non-functional Merlin, What we did see though, and that was kind of the study that Michael was talking about earlier, to our joy, if you re-express Merlin in the Schwann cells that are null, or so are not having functional Merlin, we did see a serious reduction in growth. So that gave us confidence that potentially re-expressing Merlin would be a good strategy to stop the growth of the schwannomas.
A question from Barbara Franklin. Will this approach work for all mutation types, splice site, nonsense, etc.?
So again, that's a great question from a very smart person. Good job, Barbara. So I think that's going to be part of the development. So just to reframe that question, if the goal is to replace the protein, the gene and then the protein, it assumes that the tumors lack the protein. And there are certain genetic alterations like missense alterations where that may or may not be true. And I think that the way that we're approaching this is to take, again, the most simplistic approach, instead of trying to get it in all its complexity, which is to take a situation where there is, we think, defective protein and then put the gene back in and ensure that that works, and then identify the subgroups that are most likely to respond. And there's a lot of questions about that. So to put this in the bigger perspective, to my knowledge, nobody has done gene replacement therapy in a tumor syndrome. If you think about hemophilia as an example, hemophilia is a genetic condition where people are born lacking the ability to produce certain clotting proteins that are made by the liver. And in that case, individuals with hemophilia bleed spontaneously. And historically, we've helped these individuals by providing these clotting factors back into their bloodstream through blood transfusions. And so replacing that protein was straightforward because the cells need about 7 or 10% of normal levels to have clotting ability. If you think about NF2 now, we know that individuals are born with one normal copy of the NF2 gene and one not normal copy. And tumors are the instance in where both copies, we believe, are lost in the majority of cases with little or no NF2 protein. Part of the problem is we've had technical challenges measuring protein levels exactly within cells, that is NF2 protein Merlin in cells. And so replacing, the key thing here at this stage is to show that if you can reintroduce the NF2 gene, what is the effect on tumors? Do they disappear? Do they stop growing? We don't know the answer yet, and that's the initial studies that Michael was talking about, was getting this proof of concept in place that will enable us to move it to the next level.
Okay. Question from Michael Skyer. What kinds of research do you envision occurring as a result of Youth Investigator Initiative? Strictly clinical research or more broad research on NF across the lifespan, such as a qualitative study about therapeutics?
That's for you guys, right?
Is this for the Young Investigator? So the Young Investigator Awards are normally looking at new novel idea in basic science. And this is where we are focusing instead. So basically looking at new science, developing new way to solve the problem of NF2. For instance, identifying a new pathway or identifying new modality to do it. And then the other approach we are taking is to create and the conditions to have multiple drug access the platform that Scott was mentioning. So front loading the clinical pipeline with more drugs. And this is what we need to do that is a lot of preclinical studies. And this is the other part of the, the 3 proposals that I already mentioned we are going to fund, plus, the new proposal that we are going to fund in the future.
We have 2 bergotinib questions, which I'm going to toss out here. And then we'll also just keep in mind it's 9:30. We still have most of the group still listening in. So thank you everyone for hanging in with us tonight. So we have a question from an anonymous person asking, how are participants being selected for the bergotinib trial through the NF registry, or does the site choose, or are there other ways?
Well, it's identical to any clinical trial, which is somebody has to be evaluated at a trial site. So if somebody believes that they qualify, they can call a trial site. So let's just say you live in Maine and you want to come to Mass General. I'm just using that as an example. We have patients who contact us and say, oh, I heard about this trial. Can we, be evaluated? Sometimes physicians will contact a trial site and say, I have a patient that I would like you to evaluate. So it's very open. We do want to, when the time is right and the trial is open, we'll need to make sure that we get the word out to patients and clinicians and so forth.
Question from Tawny Pendleton. Was the research completed by Synodos on the use of bergotinib published? Was tumor shrinkage shown in animals, ? In VS, ? In meningioma, ? I can't find anything on PubMed.
Yeah, that's right. So a manuscript is complete and the final edits are happening. So I would say that it should be soon to be submitted, but it does take a while from submission to acceptance. So you're correct, it's not yet published. The data has been presented in scientific forums, and there has been in mouse models seen, it depends, I have to be careful how I say this because you have to understand how the mouse models, but decreased signal associated with tumors. So a shrinkage if you go by the light emitted by certain tumors compared to untreated tumors for both meningioma and schwannoma. So as far as we can tell from the animal models, which are not perfectly predictive, very exciting data.
Question from Barbara Franklin. Are there any combo drugs you're considering, or one then the next?
At this point, we're considering one then the next, given the complexity of combining drugs. But I'm going to tell you how Scott Plotkin feels, so please take this following statement as only a personal opinion, which is for some patients, combining new drug with bevacizumab or Avastin, it's a combination I've always been interested in. We do not have formal plans to combine Avastin with any of these drugs, but for my own personal interest, not associated with this trial, I think that's going to be something that we would be interested in down the line.
And then we'll take this question, then we'll probably need to move to the next section from Tawny again. Is there a way for us to read the bergotinib research now?
To read it. I don't think that it's likely that the manuscript will be disseminated or made available until the publication, and that's because during this process it gets vetted by other professionals and usually so I don't think it's likely.
Yeah,
So
the only thing I want to mention is there is a public abstract that I think is in our conference page from last year in Paris, where this data was presented. So there is an abstract that is public, and I think that's what the editor will allow as a distribution before the full publication. This is available. And I think there are other public abstracts of presentation at scientific conferences that are available, but still, Scott is right, and the publication will not take long to come out. Annette, sorry.
Yeah, maybe one extra thing for what we are more than happy to put on the website is the Facebook Live that Jayshree Blakely did last year or a couple of months ago, specifically around brigatinib, where the data came from, what the whole process was of Synodos that has led to brigatinib, how the trial looks. But I can guarantee you, and I think we can all guarantee you, that in fact the whole Synodos project has led to a lot of discussion around drugs, drug combinations, efficacy, interpretation. So it is not, I just want to make sure that everybody understands that the fact that there is a delay in publication is not because the project has stalled. It is because there is a lot of discussion ongoing, and as we all know, scientists are opinionated, so we've had a lot of heated discussions that will only make the paper better. So it's, I understand that you want to read it now. Let's make sure, maybe Simon, we can look at, get the Facebook Live on the website so people have something to look at. Yeah. And then the abstract that Salvo.
Yep, so it is live, and we'll repost it because it's probably fallen back, as Annette pointed out. It's been a few months, so we'll repost on our blog and on social media, and we'll actually send out a link after this, again, not tonight, but in coming days, a link to this webinar, but also a link to that past brigatinib webinar so that you all can click and watch. Okay, I think, Salvo, do you want to take us to the next section?
Absolutely, yes, thank you. So thank you to Michael, Scott, for this insightful discussion. One last thing I want to add to what has been said about brigatinib is that the data is being already public through the NF data portal. So this hasn't stopped new research to be conducted on this data. The data is already been released. So if you go to the NF data portal, you log into the portal as and request access, you can actually see the whole data already available. So what we are trying to do is, if those that needs to do follow-up research on this data, this has already been possible since many months. Actually, we released it probably last summer, and so there are new proposals that are coming follow-up from the data, like the Vijaya's proposal that we're already funding. So this has not been, let's say, hampering future research. Okay, with that said, I would like to quickly move to the next section and give you just a summary of one important date. So for those of you who are listening and are interested in submitting a proposal for all of the opportunities that I was mentioning before, we have YA proposals now open for submission until January 4, 2020. So if you have a postdoc or you have a pre-doc student that you think you have a nice NF2 project you want them to work with, please apply and we'll take it into consideration. We have set aside a budget reserve for NF2, so we have bigger capacity than previous years because we want to focus on NF2 research. And then, a new cycle of DDIRR grants is now open until the end of December 31st. All details are on the research section of the ctf.org website. In January, just for you to keep it on the radar, we'll open calls for a new drug discovery initiative collaboration with Dell Open. And Dell Open is a program that stands for DNA encoded libraries in collaboration with WuXi AppTech. WuXi is the biggest CRO in the world, and they agreed to give this technology or provide the technology to applicants of DDI. So we launched this together. All details will gonna be published very soon. We announced, I think, the collaboration a few weeks ago, and details are being worked out, and this call is gonna open early January. So keep an eye open. And then throughout the 2020, we'll announce new funding opportunities. So, and with this said, I'm gonna move it to, where we stand with funding. So I would like Michelle to take over from here and guide us through.
Thank you, Salvo, and I hope everybody can hear me okay. I want to thank everybody on the phone, as all of you are investors in this project, whether you have given $1, whether you've raised $1, whether you've helped us from a marketing perspective or a social media perspective. Each and every one of you is important to our ensuring that we can move the needle for NF2 research forward. So for that, I thank you. I'm going to talk a little bit about our current funding, thank our current funders, and also let you know about what you can do. So as you can see, this project is a $2.4 million project. It was launched on World NF2 Awareness Day in 2019, which is May 22nd. We were thrilled to be able to announce this via webinar, just as we're doing this evening, and grateful that we can provide you with an update on the progress since then. We have raised about $1 million to date, and that is thanks to many of you on the phone and many of your friends and many of your family members. Our goal is to secure the remaining 1.4 million on or before World NF2 Awareness Day, May 22nd, 2020. So as Salvo has wonderfully outlined here, we have assigned 614,000 of the 1 million. We still have $386,000 to be directed, and still yet to be raised is 1.4. I'd like to talk a little bit about our major funders. Salvo, I don't know if you can click on our major funders. I'll start with, great. So our major funders, this is a snapshot of many of you on the phone tonight, not all of you, but many of you who have given generously of your time, your treasure, and your talent to help us get to that 1 million over the course of the last 6.5 months. A special shout out to the Toms family as they seed funded this project late last year, early 2019, with about $500,000, if not more, about 550,000. And we are truly grateful to them and their vision for helping to make this project happen. And as well, many of you, again, on the phone, the Morris family, Carol Harrison Gallagher, the Brainan family, the Harrison family, Peggy Wallace, who is instrumental in helping much of this research to take shape, Suzanne Earl, Tracy Galloway, who has provided a $100,000 match towards this project, the Heal family, Rick Horvitz, and his wife, Erica Hartman Horvitz, the Robbins family, and many, many more. So thank you. Now I'm going to let you know what you can do to help us in raising the awareness to help us get to that 1.4 million left to raise before May 22nd, 2020. So Salvo, I'm going to have you go back to the main slide and go to the what you can help us, what you can do. So in terms of next steps, we have created an external NF2 Accelerator Campaign Committee. I want to thank John Morris and R.B. Harrison, both of whom are on the phone this evening, and both of whom are NF2 parents, for agreeing to be the co-chairs for this committee. And there are a number of our committee members or members of our community who have agreed to be committee members as well, including Tracy Galloway, Edith Garrett, Laura and Ron Levine-Lipoff, and Roland Toms as well. So I want to thank everybody currently. The Accelerator Campaign Committee, what it will be doing is really helping us look at strategies for fundraising, but also how we can market and do outreach to the NF2 community to keep them apprised of what we were doing on the research side. I want to make sure that we're shouting from the rooftops the successes that are happening in the NF2 community. CTF successes, everybody else successes. We can't do this without you, and we need your help to make sure that we're reaching out to the community to let them know what's happening. So how can you help us do that? You can make a donation at ctf.org/endnf2. It is open to make a donation of any size, whether that is $1 or whether that is $1 million or anywhere in between. We also have the ability for our constituency to create a friends and family campaign. We will create a website for you that you can then use to send out to your friends and family to help to raise more money. You can host a Facebook fundraiser. We know these are incredibly successful. Two of our constituents just raised $1,500 to $2,000 during their birthday fundraisers. Submit your NF2 story to ctf.org, tell your story. We want to make sure that we can talk about NF2 and talk to our patients' stories. They're so inspiring and they help others know that they're not alone. We post a lot of stories and updates on our social media platforms. Make sure you're following us on Facebook and on Twitter and on Instagram and on LinkedIn, and repost what you read from us. Host an event. It could be a dinner between friends. It could be a cultivation event. It could just be an awareness event to make sure that people know about CTF, make sure they know about NF and NF2 specifically. Be one of our advocates. We have a patient engagement program, which Tracy Ann Rose leads. Talk to myself or anybody on the phone tonight, any of the staff members that you work with. Find out how you can be more involved in the patient engagement and learn how you can help us in terms of reviewing research that's being vetted. Staff a table at an NF clinic. We have a wonderful volunteer out in California who actually staffs a clinic once a month at UCLA. We can do more of that. If you're free, let us know. We'd happy to connect you with Heather on our NF clinic team and to be sure that we can work with our NF clinics to have a table. It's a great way for us to make sure that we're reaching more patients and again, ensuring that they feel as if they are with the community and with family and not alone. Again, talking to other patients and other family members, and participating in our NF Awareness Month in May. We do an amazing Shine a Light program, which is not just our walks, but they are our ability to raise awareness and make our cities and our major monuments go blue and green. If you're interested in learning more, talk to me, talk to Simon. And of course, there are so many other ways to participate and raise money for NF2 research, whether that is through a walk or a special event, or doing a DIY event, creating something of your own. The sky's the limit in terms of what we can do. Again, I want to thank you. If you've raised $1, you've given $1, you've helped us raise awareness, you're investing in this program. And as one of our major supporters said recently, I'm not a researcher. We have researchers on the program tonight who will be finding those cures. But your dollar may be the dollar that absolutely helps us find that cure. So I want to thank you. You can email me or call me. My email and my phone number is at the bottom of this slide. And I look forward to hearing from all of you on how you'd like to help and get more involved. So with that,
is there any other questions from folks? I'm sorry, Annette, go ahead.
Yeah, no, I just wanted to make one general observation. One of the biggest problems, so I was in China last year to talk about the Children's Tumor Foundation and about NF in general. There is about 500,000 patients in China that have NF. I visited the 3 or 4 clinics that see NF patients, and that was for a total of, I estimated, about 3,000 patients. What they told me there is that the biggest problem is that people do not know what NF is. And I think raising awareness and making sure that everybody knows what NF is will automatically raise our funds for all the funders that are funding NF research. But I think our biggest enemy these days is that people do not know what NF is. So shout it out over the roof that the world knows what NF is, so we can get the work done. This is my plea for the night.
If anyone has any questions, please feel free to type them in, or if anyone would actually like to speak, just let us know in the chat function, or I think there's a thing called raise your hand. You can click that, and we'll just give it a few minutes to see if anyone would like to, because I know it's easier to speak sometimes than to type in. If anyone would like to do that, please let us know. Looks like someone, hang on one second. Oh, we just got a thank you from Tawny. Thank you, Tawny. Okay, we'll give it another 30 seconds or so. John Morris, no questions. Nice job. Thank you. Thank you, John. We'll give another 25 seconds. If anyone has anything they'd like to say, or if they'd like to be unmuted, we're happy to do that. Okay.
So maybe, Simon, I received one question on how the foundation decides on which NF2 initiatives will be included or not. And that is in fact it's a great question. It is really driven by the NF research and clinical community, what we fund. So what we really, for example, for Synodos for NF2, we posted our observation that the patients were confused, that certain drugs were reported to work and other drugs were reported to not work. So we just threw that out, way of speaking, on the website and asked the research community to come up with a great idea to tackle that problem. And that is then where Scott and Jayshree and Wade and Jim Gazella came out of the woods, assembled a team to really tackle that problem. And that is what led to brigatinib and to some other drugs that are upcoming. The Young Investigator Awards, Drug Discovery Initiative are all driven by the community. Also the NF2 Gene Therapy was of course something we threw out there a couple of years ago, saying this is really working for other rare diseases. What about NF? NF1 is such a big gene that is going to be complicated for replacement therapy, but NF2 is doable. So can somebody come up with an idea? So just to say, we just throw out what we think is important, and then we let the research community throw out their best ideas. They get then subsequently peer reviewed by the best and brightest specialists in the world that can really assess and select the best initiatives, and then the studies that will be funded. So it's really a process of peer review, great initiatives, us just throwing out problems rather than answers.
Great. I'm not seeing any other questions. So with that, I think we come to the close of this webinar. Thank you everyone for participating. Thank you for being patient while we worked out that kink. So if anyone, Salvo, Scott, Annette, Michael, if anyone has anything else they'd like to close out on.
No, thank you everybody for being patient and stayed with us tonight. I hope this was useful, and looking forward to the next update.
And Michelle, anything from you? Yep, she might be muted. So, okay, anyone else?
Well, maybe one thing is that if for a next webinar, there are certain things that people really want us to talk about or tackle, please let us know, because we are just informing you of what we are doing for the moment and how we are making progress towards ending NF2, which is the obsession that I think we all have on the phone tonight. But if there are certain specific topics you would be interested to hear about, we can absolutely adapt webinars to your request.
Yes, that's excellent. Yeah, so if you have ideas, you can feel free to reach out to anyone that was on this call, or most easily, if you don't know our email address, it's info@ctf.org, and we'll make sure that your ideas, your questions, your requests get forwarded to the right folks at the foundation or as part of our partnership teams. I think that may be it. Thank you, everyone. As we said earlier, this has been recorded. In the coming days, we will send a email to you all letting you know where it's been posted, and then we would encourage you to share it with everyone in the community. As Annette said before, we need to spread the word. With that, thank you, everybody, and have a good night.